Hi, welcome to another lecture. This lecture will be about muscle tissue, and really muscle tissue is the fourth tissue we will be talking about, so let's start. It consists of elongated elements. This term is important, they're elements, and they're able to contract. Yeah? They're able to contract. And there is not much intercellular mass between these elements, but definitely more extracellular matrix than in epithelia and nerve tissue. So very small amount of extracellular matrix, but more than in epithelia and nerve tissue. These elements are connected together. There are these intercellular contacts they're working together, and these contacts are very important in order to muscle tissue to function. So we're going to learn about them. And nuclei of these elements, muscle tissue elements, are bar-shaped or cigar-like shaped. Yeah, I like this more, cigar-like shaped nuclei of muscle tissue. We have three basic types of muscle tissue. In this picture we can see the basic units of these basic types of muscle tissue, the muscle elements, basic elements. First, the striated skeletal muscle. So we're going to describe this type of muscle tissue first, and the basic unit is a muscle fiber. Basic unit of skeletal muscle tissue is a muscle fiber. That's very important because this muscle fiber, it's not a cell, it's a syncytium. Syncytium. What does this mean? It's a syncytium. It's really a multi-nucleated structure formed by the fusion of cells. So it's a multi-nucleated structure. That means that we can see many nuclei in one fiber. And that is because first we had these small cells that fuse together, create a syncytium, a multi-nucleated structure called a muscle fiber, right here. So that's the basic unit of skeletal muscle. Of course, the number of cells that converge together also varies according to size. So bigger muscle, more cells need to merge together. The small muscle, not so many cells needed to fuse together. So that depends on the size, but it can be up to thousands of cells. Just imagine how big your muscles are. But you already know that you already know muscles from anatomy, so just imagine a muscle and imagine how long they are. For example, this muscle right here, there are many of them, but I know I don't have many, but but they're very long, and so are the muscle fibers. So really the basic units are as long as the muscle itself. So muscle fibers are like this long. That's very long. Yeah. So imagine how many cells need to fuse together to form such a long muscle fiber, like a lot. Yeah, I don't know how many, but a lot. In the width, the diameter of a muscle fiber really depends on the size of the muscle, but on average it ranges between 10 to 100 micrometers. Yeah. Muscle fiber is really a big structure. We can see it in light microscope. We can see muscle fiber in light microscope, and it's really a big structure to be called a cell, because sometimes you can find the term cell in some textbooks, but rather describe this body as a muscle fiber. Don't describe it as a cell, because like is a cell long as your fucking arm? Like, no, it's not. So describe it like muscle fiber. It's best term to use for the skeletal muscle tissue. An example of another syncytium in human body. Can you figure it out? Like another syncytium in human body? Muscle fiber is not alone a syncytium, but we have these cells. I'm going to write them down. Osteoclasts. Do you remember? Osteoclasts are another example of syncytium in the human body. Yeah. So This is a common question during examination. Like, we have this muscle fiber, it's a sensation. Do you know another sensation in a human body? I don't know why they like this question, but they're asking it too often. Not to remember, so please remember osteoclasts in the bone tissue, which are eating the bone tissue, are another types, another example of sensation in the human body. Yeah. So, in other two types of muscle tissue, the basic units are cells. So, in cardiac muscle tissue and smooth muscle tissue, we have cells. Yeah? Smooth muscle cells and cardiomyocytes are also cells. So, just remember, for the skeletal muscle tissue, we have fibers, and for the other two types, we have cells, which are connected together somehow. Yeah. Let's start with skeletal muscle, skeletal striated muscle tissue, because really skeletal muscle tissue is striated, and we're going to see it, the striation. The basic unit, once again, muscle fiber, very important. These informations, all of them are very important. So this is the sensation. They are able to contract rapidly. The contraction is controlled voluntarily, so you know when you're going to stretch your muscles, so you know it, probably, I hope so. In diameter, they are about 10 to 100 micrometers. Okay, we know that. In length, they are in millimeters to tens of centimeters. So really just look at how long your muscles are, and you know how long are the fibers, or how they can be long, or whatever. And or there's something going on outside, but okay. The nuclei are situated at the periphery of fibers. Nuclei are situated at the periphery. And why is that so? Is that so because myofibrils, which are the units, smaller units of muscle fibers, these myofibrils are composed of cytoskeletal proteins, cytoskeletal structures. There are so many inside of the muscle fibers that the nuclei are pushed on the periphery. They're just basically, because of the myofibrils, the nuclei are pushed to the periphery. Yeah? So, basic things. It's a muscle fiber in skeletal muscle tissue. It's a sensation. It's about 10 to 100 micrometers thick. It's very long. It's long as the muscle itself. It contracts voluntarily, and the nuclei are situated at the periphery of fibers. Yeah. So you can recognize muscle fibers because of the nuclei that are situated on the periphery and because of the striation. And why is that so? It's because the myofilaments that are arranged in the myofibrils, there are too many of them, so the nuclei are pushed away. So that was the recap, and now let's move on for the picture from the light microscope. We have a light microscope image here once again. And here we can see cross sections. So these are the cross sections of muscle fibers right here, and longitudinal sections in the middle, which are very, very long longitudinal sections of muscle fibers. So this is how thick muscle fibers are in the middle. It's a very long formation. And at first glance, we are interested in about three things that we need to like somehow describe when we want to say, like, okay, this is the skeletal striated muscle. So we need to look at it and describe it somehow. So first, they're pink. Muscle fibers are pink. Okay, so I should say like eosinophilic, maybe that's the correct word to use in histology. So yes, they are red, they are pink, they are very eosinophilic. Dark eosinophilia is here. And the tincture of this eosinophilia is not homogeneous. So you can see it's not homogeneous. You can see this striation. There are some strips that are darker eosinophilic, some are lighter eosinophilic. And really what we can see are these transverse stripes that are result from the arrangement of the cytoskeleton. Cytoskeleton inside these muscle fibers is somehow arranged, and because of that we can see this striation in the muscle fibers. We can see it in a light microscope. Normally you're not able to see cytoskeleton, but you can see striation because of the cytoskeleton. I'm going to explain this. This is very important, and probably during your examination, somehow when there will be a skeletal muscle on the specimen, you will describe it. So it's very important to learn what it's made of, and there are these common questions during test, also during oral examination, like why is it striated and other stuff. So I will explain that. And okay, what we can see here, so these are the cross sections, and we can see the nuclei on the periphery right here. Here is the nice nucleus here. Sometimes we can see more of them because remember it's a syncytium, so sometimes we can see many nuclei on the periphery. Like here there's one, second one, third one, fourth one, and that's pretty normal. And sometimes we're not able to see any nucleus like here. It's because of the section. Yeah, but it's pretty normal. It's because of the section, but they are located on the periphery of a muscle fiber. Okay? It's a common sign that we're looking at the muscle fibers, that we're looking at the skeletal muscle tissue. Okay, so sometimes it looks like they're in the middle. In the longitudinal sections like these, sometimes it looks like these nuclei are in the middle, and you're like, wow, why is that so? Because we are in some other muscle tissue, or what's going on? And it's really just because of the section. Yeah, the plane of the section was on the periphery, so it just looks like the nucleus is in the middle, but it's not. Yeah, it's not. Just remember, look for the cross sections. It's better to look for the cross sections because here in the cross sections you can clearly see that the nuclei are on the periphery. That's the common sign how to recognize the muscle fibers. Yeah, so these are the muscle fibers, big syncytium of a cell, multinucleated structure. Yeah. And as I said, the nuclei are cigar shaped. We can see it right here in the longitudinal section. They are cigar shaped. Yeah, they are like this, cigar shaped. I was trying to draw a cigar, but never mind. Forget about this. But okay, they are cigar shaped, long, but in a cross section you can see only dots, and that's normal. Like imagine you cut a cigar, there are going to be just a little dot or like a spot like this, so it's normal. So if we look at the same specimen through polarizing microscope, that's very special type of microscope, we see that the strips light up somehow. The striation just light up, and it lights up in an opposite way. So when here in this light microscope we see some dark eosinophilic lines in light microscope, in polarizing microscope they are light. Yeah, in polarizing microscope they are light, and they are called A bands or anisotropic. Anisotropic. I'm sorry about the writing, but this is the end of my presentation, and if I click any further it will, oh no, it's functioning, but okay. Anisotropic. Anisotropic because they are light in the polarizing microscope. Yeah, they're shining, so that's why they call anisotropic, and we call it A band. So it's light in the polarizing microscope, but in light microscope it's dark eosinophilic. On the other hand, we have these I bands. Yeah, I bands. So we have A bands and I bands. These are in no, not that, isotropic, isotropic. And that means they're not shining, they are just these dark lines here, these bands, dark bands. But really, in the light microscope, these dark bands right here are in light microscope light. So these lighter bands are called I band. Yeah? So we have A band and I band. And now you know why they are called I bands and A bands, because it's because of the polarizing microscope. If they are shining here or they are not shining, it's an isotropic or isotropic. Yeah, so just remember it's because of the polarizing microscope, the names of the bands. But really, we need to describe them in light microscope. Yeah, we don't have polarizing microscope during our exam, so we need to describe them in light microscope and the basic structure. Yeah. Really, the A and I bands can be observed throughout the fiber. Here's the picture from light microscope once again. These are the fibers over here. 1, 2, 3, 4, 5, maybe 6 fibers. Right over here we can see nuclei that are located on the periphery of these fibers. And we can see this striation. This is due to the regular arrangement of the cytoskeleton. Because really myofilaments are part of the cytoskeleton. This is the smallest unit, really. Myofilaments are divided into thin filaments and thick filaments. Yeah? And the myofilaments, really the thin filaments are very similar to microfilaments that you already know. The thin filaments are just microfilaments. They have some stabilizing proteins on them, but they're very similar as microfilaments. And thick filaments are different. They are very special for muscle tissue, and they are made of myosin molecules. I'm going to explain that later. These myofilaments are together very regularly arranged, and they are arranged in the bundles called myofibrils. So myofilaments create myofibril. Myofibrils, so first we have myofilaments, then we have myofibrils, and myofibrils then form a muscle fiber. So many myofibrils together form a muscle fiber, this structure that we can see over here. Yeah? So it's like that. The diameter of this myofibril, I'm going to use another color. So here we have a myofibril, and the diameter of myofibril is 1 to 2 micrometers. So they are 1 to 2 micrometer thick. That means that we can see them in a light microscope. Yeah? So they are big enough to so we can see them in light microscope. But in electron microscope, we can see them like this. So these are the myofibrils, these structures. This is the myofibril, and we can see 1, 2, 3, 4, 5, maybe 6 here, many myofibrils that are really arranged in parallel in the muscle fiber. So we can see them as big structures in electron microscope, but we can also see them in light microscope. And really, first we're going to describe them here, because these myofibrils that are here, they contain regularly arranged myofilaments, and you can see there is some kind of striation in the myofibrils right here. You can see there are some parts that are darker, or that are more electron dense, I should say. Some parts are more electron dense, some parts are lighter, so they are striated. And these myofibrils are linked together they're like connected, just a little bit connected, not very firmly, but they are connected together with an intermediate filament called desmin. So please remember desmin as an intermediate filament that is connecting myofibrils together in parallel, and because myofibril itself is striated and they are connected in parallel somehow, not very firmly, but they are connected, we can see this striation through the whole muscle fiber, if you know what I mean. I hope so. But these myofibrils also, as I said, they are not connected very firmly, and we can see it here because what we can see here between these myofibrils, this is the cytoplasm of a muscle fiber. Cytoplasm, parts of cytoplasm with mitochondria inside. These dark, dense structures here, these are the mitochondria between myofibrils. So they are not attached to each other, they're just like interconnected, but there is also cytoplasm between them. So what am I heading to? Is that we can see also myofibrils in light microscope. We can see this striation, but we can see that myofibrils here, that the striation is kind of shifted. So we can see this is a myofibril, for example, this is another one, third one. So there is this striation going through all the muscle fiber, but it's kind of like moved to the side. Yeah, it's a little bit, it's shifted. So, and because of that, because there is some kind of cytoplasm in between, we can see the myofibrils here. Yeah, so we can see the myofibrils here in the longitudinal section, right here. And what we can see here, as I said before, myofibril is composed of myofilaments, and we differentiate myofilaments into thick and thin filaments. And what we can see here, this is not very big magnification, so we can see thick filaments over here also. Yeah, so thick filaments are these thick filaments in the myofibril. So these electron dense lines over here. I'm not sure you're able to see it, but between these electron dense lines, which are somehow thick in this picture, between them we can find thin filaments. Just have a look home at this picture very closely. Sometimes you can see also these thin filaments between thick filaments over here. Yeah, sometimes we can't see it, sometimes not, but yeah, they are there. And we're going to explain this whole structure of sarcomere is a basic unit of a myofibril. So we have this long myofibril, and this myofibril is segmented into sarcomeres because of the arrangement of thin and thick filaments inside. I'm going to explain that. And this is also telling us, this cytoplasm between myofibrils is telling us that the muscle fiber, the whole muscle fiber, isn't just a bundle of myofibrils. Please don't describe it as a bundle of myofibrils. There is also cytoplasm inside. There are also organelles, there are nuclei and other stuff. So it's not just a bundle, it's like a living structure. Yeah, it's an organ, not organ like muscle fiber itself. Muscle is an organ, but like the muscle fibers are really a syncytium. It's a created by fusion of cells. So there is also cytoplasm with organelles, with all organelles that normal cell needs for it to function. Yes, let's go for another picture of a light microscope. So here we have 3 muscle fibers over here. So first, second, third, very high magnification. Wow. Oh, you're probably not able to see this during practical trainings, so because we don't have such big magnification, but you can describe some things also, and you can see some things also during practical trainings. So once again, we see sections of myofibrils. It's a little bit like it's a little shifting. I'm sorry. So a little shifting. These are the myofibrils of a muscle fiber, so we can see them because of the shifting. Once again, I'm repeating myself, I'm sorry. And we can see nuclei also. So here we can see a nucleus. I'm going to just with green because it belongs to the muscle fibers. So this is the nucleus, cigar-shaped nuclei on the periphery of the muscle fiber, and here we have them once again. Okay, but there is also between these muscle fibers, there is this extracellular mass also. So there is some kind of space. This is extracellular mass, and this nucleus right here probably belongs to this extracellular mass, and this is probably a nucleus of a fibroblast or a fibrocyte, probably fibrocyte, because I can see there's this condensated nucleus that's not very active, so probably it's a fibrocyte. So there is extracellular mass, really, which is produced by the muscle fiber itself, but also by a fibroblast that are in this extracellular mass. We should call it probably extra fiber mass because it's between fibers, not between cells themselves, but it doesn't matter. Okay, so but just remember that there is also some extracellular mass in the muscle between muscle fibers. Yeah, and here we can see this striation. So let's go back. Where more details can be seen here. In the middle of an isotropic band, remember isotropic band is this light band over here. It's isotropic band. It was dark in a polarizing microscope, but in this light microscope it's light. So these are isotropic bands, and in the middle of each isotropic band, we can see a line, a dark eosinophilic line. Everything is that eosinophilic here in this muscle fiber except nucleus, but we can see this dark eosinophilic line in the middle, and this dark eosinophilic line in the middle of each I band is called Z band, or Z line, I should say. It's not really a band, it's just a line, so it's called the Z line. Yeah, so these are the Z lines right here. And also in the middle of an isotropic band, which are these darker eosinophilic bands right here. These are A bands, simply A bands. And in the middle of A bands, we can see a less conspicuous, lighter line. Also, I know you're probably struggling right now, but also there is a light line in the middle of A bands, and these light lines are called H lines or H bands. So there is also a light eosinophilic line in the dark A band, and there is also a dark eosinophilic line in a light I band. I hope I said it correctly. If not, I'm sorry, but you can see it. Yeah, you can really see it. And this is the basic structure. And basic unit of one myofibril over here. Yeah, basic unit of one myofibril is a sarcomere, and sarcomere is a unit that is from one Z line to another Z line. So this is one sarcomere, and this is next sarcomere, and next. So we have one Z line, second Z line, and everything in the middle is called sarcomere. That's why it's called Z line, because it's from German. It means zwischen. That's between something or something. Okay, so it's switching a bit, it's between sarcomeres, like the Z line. It's because of that. So, and we will describe this in more, even more details, so be prepared. In the cross section, this is a cross section of muscle fibers. So once again, we have these muscle fibers. Some of them are lighter, as this one. As you can see, that's not an artifact, that's normal. We get to that. So these are the muscle fibers. We can see the cross sections. We can see the nuclei on the periphery. Yeah, the cross sections of nuclei on the periphery, right here. Very well, very good. Basophilic nuclei. And what we can see also are these eosinophilic dots over here. Cross sections of eosinophilic dots over here. And these are myofibrils. These are the cross sections of myofibrils. So fibrils, we can see cross sections of myofibrils in a cross section of a muscle fiber. Yes, so it's pretty normal. They create these fields in cross section. We can see these fields in cross section, and they are called Cohnheim's fields, like this. So they are just like arranged together in some fields like this. You can see it. There is more mass of cytoplasm, more cytoplasm between this bundle of myofibrils right here. So it's arranged into fields, and these fields are called Cohnheim's fields. Sometimes you can read about them in some textbooks. So I'm just writing it down, Cohnheim's fields. So they look like this, just like bundles of myofibrils, I guess. So this is the normal look of the cross section of muscle fiber really look like this. Also, once again, it's very big magnification, so you can see it clearly right here. Well, and the sarcomere and the basic unit of myofibril, well, what did I wanted to say? It's very difficult to describe it. We can see once again the myofibril is made of thick and thin filaments. Yeah, the thick and thin filaments. The thick filaments, which are here, I'm going to label it with blue. These are the thick filaments, are in the middle of sarcomere. In the middle of sarcomere, right here. And really, okay, so we have this thick myofilaments in the middle, and we have two bundles, or two sets, I should say, two sets of thin filaments on the periphery. Thin over here. So here we have the Z line, and there are these thin filaments that are connected to the Z line, but really we can see only half of them because they are, you can't even see them here because they're like too thin, but we can see them like this, in this whole, like crossing the whole eye band over here. So this is normal location of thin filaments right here. They partially overlap, these thin and thick filaments, you can see it right here, they partially overlap, but also we see them separately. We see a section where we can see only thin filaments, and we see section where we can see only thick filaments, yeah, but they are partially overlap. And it depends on the contraction, if they overlap more or less, so really it depends. Thin myofilaments, these thin myofilaments, yeah, these thin myofilaments are attached to the actin binding proteins, that is alpha actinin, that's actin binding protein, and they bind to the region of this Z line over here. Yes, Z line. So because of alpha actinin, they can bind to the Z line, the region of Z line. So once again, at the periphery of each sarcomere, we have these two Z lines which like intersect a strip in which the myofilaments are thin. Yeah, so there is this strip or a band, the I band, as I said, and the Z line is in the middle and is like connecting these thin filaments into the Z line, which are crossing through the I band. Yeah, so that's how it looks like, really. And so we can see only half of I band over here, I band. And the entire middle area where the myofilaments overlap here, I'm going to take another color for this. So here we can see, no, that wasn't working. So here we can see the thin and thick filament overlap. Yeah, so that's this structure over here, this band, and this is A band. A band. And there are many questions in examination, during examination, like what can we find in A band? And the correct answer is, well, thick filaments and also thin filaments, because in this A band they are overlap and we can see both of them. But what we can see in I band, in isotropic band, only thin filaments. We can see only thin filaments in I band. So sometimes you have these pictures. These are like cross sections of filaments in the sarcomere, and in this section you can only see, for example, thick filaments. In this section you can see thick and also thin filaments, so you know this is an A band. You know you can see here only thin filaments, and this is the I band. And here we recognize also H band right here. And in H band, there are only thick filaments in the center. And when the sarcomere contracts, it really just pushes thin filaments to the center. So when there is a contraction in the muscle, the H band will disappear. Yeah, H band will disappear, and we're not able to see H band. So, and I'm going to do that later once again. So just remember right now, we have these bands. This light band in a light microscope is called isotropic I band, and it only contains thin filaments. Thin filaments, these are just like microfilaments that are connected to Z lines. Yeah, Z lines. Then we have this A band that is an isotropic band that's really dark. Yeah, in light microscopy. It's light in a polarizing microscope, but it's dark in light microscopy because we have also thick filaments and thin filaments together in one band. So that's why it's darker. That's why it's darker eosinophilic, because there are more proteins in it. Yeah, so you should know there are also thick and thin filaments in A band. And also sometimes we can describe this H band in the middle of A band that contains only thick filaments. Yeah, but this depends on the state of contraction. And one last thing, it's written here, mesophragm, and it's M line. So in this H band over here, H band, we can sometimes describe M line in the middle. It's just a thickening of thick filaments. In the middle of the thick filaments just like thickened, they're more wide. So sometimes we can see this M band or M line right here. Just a thickening of thick filaments. Wow, that was complicated. but I hope you can understand this. This is very important stuff. You should be able to describe all bands. These are the common questions during exam, and also you should be able to describe them on electron microscope, because we have pictures from skeletal muscle, pictures from cardiac muscle, and this is the same as in the cardiac muscle, for example. So you should know how to describe this striation, these bands and stuff, and what's happening here. It's because of the cytoskeleton and what we can find in the muscle fiber. So once again, remember, this is the picture from electron microscope, and once again we can see these myofibrils, many myofibrils over here, probably 6 of them. Yeah, so they're pretty thick, 1 to 2 micrometers. We can see them in light microscope also. And now we can see these bands also in the electron microscope. So once again we have, I'm probably not able to use the same colors as I used before because I don't remember them, but you should understand it. Yes, so we have this basic unit that is a sarcomere, which have Z lines, one Z line, this electron dense line here, and this electron dense line here, that's a Z line. Yeah, Z line. And there are thin filaments that are connected to the Z line over here, and really we're not able to see them properly because they're so thin, but you should know these thin lines are, thin filaments are like this. Okay, so these are the thin filaments going like this between thick filaments also in here, so it's looking just like this. And this band, which contains only thin filaments, is called I band, isotropic band. This band with only thin filaments. You can see they're so thin we're not able to see them properly. Then we have this A band, which contains also thick and thin filaments together. So we can see these thick electron dense lines, and these are the thick filaments. Yeah, so the band, A band, contains also thick and thin filaments, is in the middle. Then we have H band sometimes, but it's like very hard to see H band. Probably it's just like this part over here. I can see it's lighter, yeah, lighter part in the middle, and there is also some kind of darker part in this lighter part in the middle, and this probably will be the mesophragm or the M line, but that's too much, probably too much. It's hard to describe it sometimes. It's more of like imagination, and it takes a lot of training to see this, but you can see Z lines. You can see how sarcomeres are organized, and you can see thick filaments and thin filaments together creating A band. Yeah. So, and just remember there is a cytoplasm also in between with mitochondria and stuff. So, and these Z lines are interconnected together with desmin. Yeah. And that's why we can see the striation throughout the whole muscle fiber. This is the cross section of myofibril. Remember these small eosinophilic dots in the light microscope? Well, now we can see the whole big structure. This is a cross section of myofibril, and what we can see right here. Okay, so there is a cytoplasm also between these myofibrils. Yes, there are probably some mitochondria or other organelles. I can't really tell based on this picture because it's not so clear, but we can see cross section of myofibril, and once again we can see these bigger electron dense dots. These are the cross sections of thick filaments over here, and we can see also electron dense dots, but smaller ones, smaller electron dense dots, and there are really 6 of them around one thick filament, and these are the thin filaments, thin filaments over here. So 6 of them around one thick filament. Yeah, but it cannot be said that the ratio would be 1 to 6. Yeah, because it's really not like that, because those thin filaments are shared between these thick filaments. So the correct ratio is rather 1 to 3 or 1 to 2, 1 to 3. 1 to 3 probably. And really this arrangement, you can see this arrangement is so regular. You can see it's so regular. It's sometimes called a paracrystalline. It's like crystal. Yeah, so it's described as a crystalline structure, and that's because it's so nice. Once again, we can see very big magnification of the sarcomere, myofibril. Here is the nucleus. It's somehow like strange picture for me, but we can see clearly a sarcomere as a basic unit. Once again, Z lines over here. They're calling it Z band, but it's better to call them Z lines because they're really lines. And around them there are parts where there are only thin filaments over here. This is the I band. But thin filaments are also going this way to the between the thick filaments, and this is the A band, which contains also thick and thin filaments. In the middle we have H band, which contains only thick filaments. In the middle of H band we have M band or M line, mesophragm, which is a thickening of thick filaments. Everything is connected together nice and firmly, and myofibrils are also connected together via desmin. Yeah, so you should be able to describe it like this. So, and we have some molecular structures, really, here. Not like molecular structure, but thin filaments and thick filaments are composed of something, and you should know like how the structure of thick filament and thin filament really looks like on a molecular level. So let's start with thin filaments. They really resemble microfilaments. This is the thin filaments. It's basically the same structure. Yeah. It consists of 2 helices of F-actin, which consists of globular parts, yeah, G-actin. This is the basic structure. You should describe it also in the microfilament of the cytoskeleton of basic cell, but also there is this thin filament in muscle tissue. You should know it. It's somehow different. It's 8 nanometers thick. So it's thicker, a little bit thicker than a normal microfilament in normal cells. In this case, the actin filament is stabilized. It's stabilized. If not stabilized, it would tend to disintegrate, depolymerize, and you don't want that in the muscle tissue. You want the contraction, you want everything to work, so you need to stabilize the actin filaments or these thin filaments, really. And the stabilization molecule which is used here is a tropomyosin. Tropomyosin is also a double helix. You can see it right here as green structure. It's a double helix. Once again, like, what's up with the double helixes in nature? Like, everything is somehow twisted together with double helix, like DNA, microfilament, and tropomyosin also. I think nature really likes double helixes when they're 2 structures that are somehow twisted together. But okay, I like it. Never mind. Tropomyosin is really a protein that occupies 7 globular heads of G-actin. So it sits on the 7 globular heads. 7 heads, not human heads, but heads of G-actin. And there is this troponin complex that is also attached to the tropomyosin. So these are the proteins, basic proteins, tropomyosin, double helix protein occupying 7 heads. And also there is a troponin complex. It's a complex. It's not just one troponin, there are 3 of them. This is just complex that is attached to the tropomyosin. Tropomyosin right here. So we differentiate, as I said, we differentiate 3 troponins, and that is troponin T. I shouldn't write it right like this probably. I don't know. Troponin T, troponin I, and troponin C. Yeah? It's probably like labeled like this. Yeah. Troponin T, T and T. T and Y and T and C, these are the basic 3 units of troponin complex, and they have some functions. Troponin T is probably this guy over here, which binds to tropomyosin. Troponin I is called I because it had such conformation at rest that it inhibits the binding of myosin and actin. Because basic rule of contraction is to myosin and actin to bind together, to connect actin and myosin. And troponin E in rest state has this conformation that it inhibits binding of myosin into onto actin filaments. So I'm going to write it down, inhibits. Yeah, so it has this special conformation. And troponin C has the ability to bind calcium. Troponin C is for calcium ions over here. The filament is stabilized by tropomyosin and can be activated by troponin and what's the activation really? What's happening? It's like the calcium is needed to be like calcium will bind to troponin C. In that order, the troponin I will change conformation, and the tropomyosin will be drawn deeply to the actin filament, so the myosin can bind to actin. Probably I'm going to explain it once again because this mechanism of contraction is very important. But just remember right now that they have some functions. They are important because you can measure the levels of troponin, for example, during myocardial infarction. So you should know why we have a troponin, like what's that, yeah? And we have this tropomyosin, and because of these complexes that are bounded to actin filament, the thin filament is thicker than normal filament. It's stabilized. This stabilization is very important, yeah? Length is regular, like 1.27 micrometers, okay, like whatever. And then we have thick myofilaments, which are thicker, and we can clearly see it because it's written here that these myosin thick filaments are 15 nanometers thick. Yeah, so they're thicker, and they have completely different structure, They are formed by a macromolecule of myosin. So myosin molecule is a basic structure of a thick filament, and each myosin subunit is made up of 2 heavy chains that are twisted together. So we have this big myosin molecule, and this myosin molecule also have some subunits. So we have, once again, twisted 2 heavy chains together, which have these globular heads at the end. So this is the basic units over here. But also there are light chains, 4 light chains that are connected to these globular heads like this. There are 2 on one and 2 on other one. So we have 2 heavy chains and 4 light chains together. They are creating myosin molecule, And this myosin molecule have two heads, which have two sides. One is actin binding site, because it needs to be bound to actin, obviously. And one has ATP and ATPase binding site, because we need energy to do so. We need energy to bind to actin. We need energy to contract our muscles. So we have actin binding site and ATPase ATP binding site. Yeah, ATP binding site, which has also ATPase activity, because you need to somehow cleave the ATP in order to release energy. That's what is happening here. And you can see this, there are many myosin molecules that are arranged together in this very big thick filament, the twisted structure. Yeah. And we can see these heavy chains are in the middle and the heads are on the periphery, really. And this is like a bipolar structure. Yeah, this is a bipolar organization of this myosin, this thick filament, because we can see heads on the one side and heads on the other side. Here we can once again see the construction of microfilaments, not only microfilaments but normal thin filaments. Yeah, so once again globular parts and this tropomyosin and troponin complex made of 3 troponins, the subunits, 3 subunits, double helix of tropomyosin, which is connected to 7 heads or has 7 globular parts once again, and these are the subunits. So this is how it really looks like. It's long enough to be seen in light microscopy, but it's not thick enough. It's only 8 nanometers thick. So remember, you're not able to see it in light microscope, like no way. Not even in electron microscope. Yeah, you saw that before. We're not able to see it in electron microscope because it's too thin. You know, sometimes we can see it, but not in the light microscope for sure. And these are the myosins. Myosins, like myosin molecules, are normally located in many cells. This is not just something that is located in a muscle tissue. Yeah, remember that many cells have myosin molecules because somehow they need to move things. Myosin is attached to the microfilament, so we can transport stuff in the cell. But only in muscle tissue they are organized in these big filaments, these big structures. Once again, you can see this myosin molecule right here, which is made of 2 heavy chains that are twisted together with globular heads, and in red we have these light chains which are attached to the heads of this thing right here. Of this myosin molecule is made of these 6 chains, but they're attached to the globular head. Yeah, I should say that. It's organized in this structure right here. And really, this is typical organization for the skeletal and cardiac muscles also, that the heads are on the periphery. It's like a bipolar organization, bipolar oriented, as I said before. Yeah, there are some numbers describing this. You should know probably it's like 1060 nanometers long, this thick filament. Yeah. This number isn't that important, 200 nanometers. The heads are away from each other 14 nanometers. Yeah, okay. Fine. This is a picture that I don't understand really. Hinges? This like the thick filament? This is the myosin right here probably. Okay, I'm not prepared for this presentation, but I think it's a myosin molecule. No, these are the heavy chains with these globular heads right here. Isn't that the same? Maybe I'm wrong, but I don't know. Oh, it looks like the myosin molecule. Okay, and here we have one globular head, which is like 5 nanometers thick, okay, and 16 nanometers long. And we have this ATP binding site, with ATPase activity. Remember that, ATPase activity. And we have actin binding site. These are two sites you should know, you should describe them. Why do we have ATP binding site? Because we need energy. Why do we have actin binding site? Well, because we need to bind to actin somehow, because that's the basics of contraction, to bind actin in some way. And this is once again the sarcomere. Sarcomere with thin filaments and also thick filaments in the middle. And really, in order for those filaments to be arranged so regularly, like we can see in sarcomere, in paracrystalline structure, they must be helper proteins there. It's just like they cannot do by themselves. There need to be helper proteins in myofibrils. And in the Z line, for example, in Z line, there is an alpha actinin, as we said, alpha actinin. So in Z line, there is alpha actinin, and also this desmin, which is interconnecting myofibrils, but okay, and also vinculin. You should know also vinculin. These two, you should know these two, yeah, alpha actinin, vinculin, which are connected these thin filaments to Z line. And also we have these special proteins called titin. It's a funny name, like titin. It's just a little bit twisted. And this titin is connecting thick filaments to the Z line right here. Yeah, and it's able to shorten and elongate because it's twisted, so it's able to do stuff. It's very flexible, and that's why also sarcomere is flexible. You can contract sarcomere. You can see it here. This is the stretch sarcomere where everything is away from each other, and then we have contraction. That is everything together. This is a relaxed state when it's back in the normal position, but when you contract, everything is even more connected together. Yeah, in state of contraction, Z lines are somewhere here, okay, and everything is moved together. Okay, and so the titin is important, that is connecting thick filaments to the Z line. And also we have nebulin. Once again. So, not once again, you probably heard about it first time, but we have also nebulin, which is stabilizing or providing these thin filaments over here. Yeah, so it's for the thin filaments. Supporting the thin filaments right here, nebulin. So once again, like when the contraction occurs, just imagine when the contraction occurs, there is a shift of thin filaments between thick filaments. Yeah? And this will shorten the entire sarcomere. Entire sarcomere will shorten, and this, when sarcomere is shortened, like imagine there are many sarcomeres in one myofibril and each sarcomere will shorten, then the myofibril will also shorten. Yeah, so the myofibril will shorten, and also sarcomeres of myofibrils are attached to the membrane of muscle fiber, and that means when the myofibrils shorten, also the muscle fiber will shorten. And when the muscle fiber will shorten, well, the whole muscle will shorten. So when you see the contraction, it's like the shortening of a muscle. And just imagine right now what's happening. We can see that it's shortening of a muscle fiber. And why is that so? What's happening really? Yeah? And why is that shortening? It's because these myosin chains right here, these heads, bind to actin right here, and these heads are moving on actin filaments. Yeah, that's the mechanism of contraction. So, but once again, here we have a picture of muscle fiber. And here we have nucleus, yeah, so we are somewhere in the periphery. Yeah, here we have a nucleus. These are the mitochondria, because we can see two membranes and the crysts. So mitochondria over here. There is also cytoplasm in here between the nucleus, and here we can see cytoplasm right here. And this structure right here, you already know, this is the myofibril over here. And each muscle element, like that's for the muscle fiber, but also for the cardiomyocyte and smooth muscle cell, they are covered with something called superficial lamina or outer lamina. And you already know that that's just the lamina densa and lamina lucida together. It's just like basal lamina, but it's not basally, because we are not looking at the epithelial cell, but it's all around the cell, and it's called superficial lamina. Superficial lamina, it should be somewhere here, but really I cannot see it properly. I'm not able to divide this superficial lamina in lamina lucida and densa. It's hard to see it right here, but it's there. And it's also around adipocytes and Schwann cells and what other cells? I forget right now, but also it's around some cells of proper connective tissue, like these adipocytes and Schwann cells. You know them, there are these glial cells in peripheral nervous systems, which are myelinating nerves. But also remember, all muscle elements, fibers, cardiomyocytes, smooth muscle cells are covered with this superficial lamina, which is connecting them to the outer layers of more layers that are around, which we are going to talk about. But yeah, just remember that now there is a superficial lamina over here. And here we have another picture. Yeah, once again, I don't know what should we describe here. Maybe this mitochondria over here. Once again, there are many mitochondria in muscle tissue because we need energy for that. Here we can see a part of a nucleus over here. And these are the myofibrils. So when you have picture like this during exam, you should be able to describe once again this thick filament and thin filament in between. This is the I band, this is the A band over here, and there is also a cytoplasm that can be seen here. And this is a granular endoplasmic reticulum with ribosomes attached to it, but there are many free polyribosomes over here. So, and once again, granular endoplasmic reticulum, and I don't really know what else can I see here. Probably nothing more. Once again, mitochondria. These are very looking strange, but these are mitochondria, and also there's some superficial space right here, superficial lamina probably. We can see right here there's a lamina lucida, this small, oh sorry, this is the lamina lucida, small layer, and lamina densa, but just a small place right here. But they are asking this during exam, like, what is this little shit around here, like, what's that? And you're like, oh, I don't know. But I sometimes don't know either. But yeah, that sometimes happens because we can see nucleus in a muscle cell, so it's probably a skeletal muscle. We don't really know. It might be a cardiac muscle. I'm going to show you pictures from cardiac muscle. And there is also this superficial lamina. Yeah, but it's like really there are these ribosomes and granular endoplasmic reticulum. But remember, these proteins, these microfilaments, thick filaments, and everything like these filaments in the cell are stabilized. So it's not very big turnover, maybe when you're working out too much, but they're stabilized. So it's not just like in normal cells, they're polymerizing and stuff. They're just stabilized, and that's not so much turnover. So really, based on this picture, you will probably think of why isn't the fiber basophilic? Because it contains a lot of ribosomes. I can see them. Why isn't it basophilic? Well, you're right, but there is more eosinophilia because in general there are more proteins and more mitochondria than ribosomes. Yeah, so you just remember it like this. There are also ribosomes, but they are not basophilic fibers because there are more mitochondria and proteins themselves, like these thick and thin filaments, which are creating fibrils, myofibrils. But also, I forgot to tell you, we have also this protein you probably know already. It's called myoglobin. Myoglobin is like hemoglobin, and you know, erythrocytes, the red blood cells, are very much red, eosinophilic, and this protein, the myoglobin, we can't see we can't see the protein here, but it's because it's soluble. It's soluble, so it's not forming very well seen structures, but it's eosinophilic also. So these mitochondria, proteins, and myoglobin, a special protein that binds oxygen in the muscle tissue, it's also eosinophilic. Yeah. Well, the sarcoplasmic reticulum. Okay, so sarco means meat, really. Sarco means meat, and that's why sometimes we're not just telling the muscle fiber has a membrane, cytoplasmic membrane around, but we call this membrane a sarcolemma. Yeah, and sarcoplasmic reticulum is just smooth endoplasmic reticulum. SER. Smooth endoplasmic reticulum, just another name because sarco means meat. Oops, I'm sorry. Sarco is meat. Yeah? Meat. I don't know if you recognize this is the meat, but it's meat. Yeah? It's a bone in the middle and it's meat. Okay. So, it's arranged very specifically in the muscle fiber. So here we have myofibrils. These are the myofibrils. You can see these thick filaments inside. There are also thin filaments, but these are the myofibrils. This is the basic structure of a sarcomere. Yes, so you have a look on these IA bands and Z discs or lines, Z lines over here, H band over here, and around these myofibrils, as I told you, there is a cytoplasm with organelles, and around these myofibrils there is also smooth endoplasmic reticulum called sarcoplasmic reticulum, and it's very, very specifically organized in the muscle tissue, in the skeletal muscle tissue. It's organized that it's a network of tubules. It's a network of tubules, like that's normal for smooth endoplasmic reticulum. It's a network of tubules. But here in a skeletal muscle, these tubules are fusing together and creating this terminal cisterna or tank, terminal tank here. You can see this big structure. Yeah, it's forming this terminal tank over here, and really what's happening here, I'm lost right now what I wanted to say. This terminal cisterna, the terminal cisterna is located at the interface of A band and I band, right here. It's in this interface. Yeah. And we can see there is some orange structure in the middle. So we have one terminal cisterna on one side. So one terminal cisterna on one side, the second one on the other side of this orange structure that is not normally orange, but it's in the purpose of this picture. But this structure right here, which is called transverse tubule or T tubule in short. Yeah, T tubule. Transverse tubule because it's crossing transversely. And the T tubule, the transverse tubule, is invagination of the membrane that's around the muscle fiber of the sarcolemma, of the plasma membrane. So imagine there is a plasma membrane around which is making those very, very deep invaginations. These deep invaginations are going between the myofibrils to the center of muscle fiber. Yeah, very deep invaginations. And the smooth endoplasmic reticulum is creating these terminal cisternae that are around these deep invaginations of sarcolemma. So this is very typical organization for the skeletal muscle. There is this structure is called a triad, and you know why, because we have three things connected together, triad, and we can find this triad on the interface of I band and A band, and these are common questions in during exam. Where is triad located? What it's made of? Why is that so? Right now you probably don't know the function, but I will explain. You should describe what's happening here. Why do we need T-tubule in a skeletal muscle? Why we need these invaginations? Yeah, that's very important. And this organization of sarcoplasmic reticulum, probably now you are thinking, why do we need so much sarcoplasmic reticulum in the skeletal muscle tissue? And this organization, why is that so? Yeah, so we can see this triad also in electron microscope, and we have also pictures of this triad, maybe 2 pictures of electron microscope during oral exams. You can somehow meet with it. Yeah, so we have once again a myofibril 1. The second one here is coming somewhere else. It's not sectioned very properly, but here we have this triad over here. In the middle, this part right here, this is the T-tubule, the transverse tubule, this space right here. Yeah, we can see it only in somewhere here because it's a little bit curved, so the section isn't very you cannot see the whole triad moving across because it's like moved. This is just a section, just remember that. We can see only a part of it. And really this is the T-tubule, and around the T-tubule we have a terminal cisternae. So here we have one terminal cisternae and the second one here. And this is the smooth endoplasmic reticulum on one side and on the other side. And we can see it's on the interface of I and A band over here, because this is the Z line over here, Z line. Yeah, this is the I band, probably in that case. And here will be the A band over here. So it's in this interface of these bands. Okay, so it's very important to describe this structure. Here we have a cytoplasm. There are ribosomes and stuff, so it's like that. And this is for you to like as you saw before on the second picture of cross sections of muscle fibers, you saw that some of them are more eosinophilic, some of them are less eosinophilic. And this is a special staining for succinate dihydrogenase. I'm sorry, I'm struggling, but it's an enzyme that is normally located in the mitochondria. So when we stain this enzyme that is in mitochondria, we're really visualizing how many mitochondria are in the muscle fibers. And we differentiate muscle fibers. They are more they are slow, they are more red, they're aerobic. They contain more myoglobin, which they are aerobic, so the myoglobin is binding oxygen, so they contain more myoglobin. They also contain more mitochondria. Yeah, so these muscle fibers, red muscle fibers, oxidative, so one type. These fibers are more dark in here because really they contain more succinate dihydrogenase. I tried it once again, that didn't work, never mind. And yeah, they're more red because they contain more myoglobin, more mitochondria. Not red in this case because they're blue, more blue, but in a normal state. And then we have these glycolytic fibers. They are fast, they are white, they are anaerobic, so they don't contain so much mitochondria and myoglobin, but they are very fast, really, for quick contractions. But they are quickly depleted. Quickly depleted, so they are very fast to contract, and bam bam, but they are quickly depleted, and it would not be very suitable for the respiratory system, for example. So we can find these slow red muscle fibers in the respiratory system, or some muscles that are contracting all day long, like the muscles of a spinal cord, for example. And once again, it would not be very suitable to have these glycolytic muscle fibers in respiratory and spinal muscles, because at the end of the day, we would be knocked to the ground, and we would have apnea breaks and stuff. So I'm glad that we have oxidative muscle fibers in our respiratory system and the muscle system of the spinal cord. Yeah, so they're working all day, all night. And remember, there is always some kind of combination in muscles. So when we talk about the red muscles, for example, these respiratory muscles, there are always some white fibers in these muscles. Yeah, it's a combination, but more muscle fibers are red, aerobic, slow, oxidative. Yes, so there is always some kind of combination. You can always find also glycolytic muscle fibers and oxidative muscle fibers in one place. It really depends on which one is more prominent in that muscle. So we have more these glycolytic muscle fibers, for example, in biceps or triceps or quadriceps on legs and stuff. Yeah, so but there we can find the combination always, so remember that. So this is very important because as I told you before, the myofibrils need to be connected to the sarcolemma, to the membrane of a muscle fiber. They need to be connected because if they would not be connected, the myofibrils, the muscle fiber would not stretch, would not contract also. Yeah, so we have a muscle fiber, it's a big structure right here, and we have these myofibrils inside like this, and they really need to be connected. Yeah, they need to be connected to the membrane, because if they're not connected, they would stretch, and the muscle fiber would not stretch, and that will be worthless. Yeah, worthless contraction. So they need to be connected. And why are we telling you this? Because now you can see oh my god, another proteins, another I'm sorry, what I need to learn all of this stuff, why? And you can see the thin filament over here of a myofibril is connected with a special protein called the dystrophin. This is a very special protein, to the sarcolemma, which is a membrane of myofibril, as I said, and this whole complex is also connected to the superficial lamina that is around the muscle fiber, superficial lamina here. So there are many proteins in here. The most important protein is dystrophin. The whole structure, this connection is called costomere. Yeah, costomere. The whole structure made of many proteins. And most important is dystrophin, because its gene, the gene that codes for this protein, this dystrophin, is located on a sex chromosome, X. X chromosome is located here, this gene. And if that gene is broken, if you don't have dystrophin in your costomeres, in these connection things that are very important, it's a disease called Duchenne dystrophy. You probably heard about this very, very bad disease, Duchenne dystrophy. There's no cure right now, actually. Probably gene therapy would help, but it's very hard to develop to transform this gene to create dystrophin that is defected in Duchenne dystrophy. And it affects boys normally because boys have only one X chromosome. Yeah, boys are XY. That's why when the X is broken somehow, there is no other X to fix it. Yeah, so this is an X recessive disease, the Duchenne dystrophy, and boys are affected. And it's very hard because they're dying in young age because they lack dystrophin, so their muscles are breaking down. There is no right connection, which is very important for the contraction and also for repairment and regeneration and some stuff. So it's very hard disease to begin with in life. Yeah, so remember this, Castamira. You should know during pathology, like when we're going to talk about dystrophin, like where is it located? Why is it important? So this is why it's important. It's connecting fibrils to superficial lamina. And also muscle fibers need to somehow connect to the tendons, because you know that what is going to the bone is a tendon. Yeah, so we have muscle fibers and we have a tendon that is attached to the bone. So what we can see is here that the tendon is attached to the muscle, so that the collagen fibers of the tendon are anchored. They are anchored to outer lamina or this superficial lamina. So this superficial lamina is also very important for the tendon to anchor to the muscle. What we can see here, there is a muscle fiber that is creating these invaginations, or I should say maybe the protrusions or invaginations, doesn't matter, but we can see the membrane of a muscle fiber like here. It's wavy, it's very wavy, in order to tendon to really connect, yeah, to connect with this muscle fiber right here. And this tendon, these collagen fibers are attached to the superficial lamina. Yeah, so you should just know it's wavy. It's not like straight line that is connecting. It's a very firm connection between these two. And its superficial lamina is very important in this case. Once again. Okay, this is how it looks like. This is what we are talking about like all the time right here. We can see muscle fiber as a basic unit that can be seen in light microscopy, which is made of myofibrils right here. Muscle fiber, as we said, has this superficial lamina, and also the muscle fiber has reticular fibers around, reticular fibers. So there is this superficial lamina around with reticular fibers, and the superficial lamina with reticular fibers is creating something called endomysium. So superficial lamina plus collagen type 3. These are the reticular fibers, really, collagen type 3. It's creating endomysium. So each muscle fiber is covered with endomysium. Muscle fibers together create a whole bundle of muscle fibers which have a perimysium around. Perimysium is another like a fibrous thicker sheath around, yeah? So it's made of dense connective tissue, and it's a thicker fibrous sheath that is surrounding many muscle fibers. And you can see these bundles of muscle fibers are also connected somehow together, and we can see arteries and veins and nerves that are protruding between these bundles of muscle fibers, and they are connected together with epimysium. Yeah, epiphysium, epimysium, and this epimysium is like, there is also loose connective tissue which contains these arteries and other stuff, because the muscle itself is whole organ, so you can find many structures there. You can find connective tissue there. There is not just like a muscle is made of muscle tissue. Don't tell that during exam. It's not like it's only muscle tissue. It's also connective tissue. You can see it right here. It's also nerve tissue there because we can find nerves there. It's also vascular system here. Yeah, so there are many things here that can be found, and they're covered, these bundles are covered with epimysium, which is under fascia, deep fascia. Yeah, then we have this tendon with this wavy thing. Oh, my pen is broken. It's doing this all the time during this presentation. I don't know what's happening, but okay, we can move on probably. So we have endomysium, perimysium, and epimysium. And then is a fascia. On the top you will see a fascia, probably when we look at the muscles, and the tendon right here that is attached to the bone. This is the whole structure. Well, and the basic function of the muscle tissue is contractility. Yeah, contractility once again is an interaction of actin and myosin. They are connecting together, actin and myosin. They tend to form filaments. Yes, we know that. We describe the filaments very well. So thin and thick myofilaments. Thin are made of actin, and thick myofilaments are those made of myosin. And we can see it here on this picture. It's not a very nice picture, but whatever. This is the thick myofilament over here because it's thick, and once again a thick myofilament over here, and we can see an actin myofilaments which are thinner. They are about 8 nanometers thin. These are 15, these are 8 nanometers thick, and we can see there are some kind of connections between them. You can see these bridges. These bridges are made of mostly myosin heads, because myosin heads are those that are connecting to the actin. So these bridges are made of myosin heads. So really myosin heads are attached to the actin and they are moving towards actin. So contractility. This is just a temporary interaction between actin and myosin. They can move, these heads can move because of the release of energy from ATP that is on the head, because we know the head contains ATP binding site, just remember that. And really these connections between myosin and actin are basics of contractility of muscle tissue. But what's happening here? It's much more difficult than it sounds like. Like you're probably not surprised in this state of presentation that we're going to describe the contraction in more details. So what's happening right here is that we need to activate actin first in order to contraction to happen. So myosin binds to actin when the thin filament is activated. Here's the thin filament, this is the thin filament with the myosin head, and we need to activate the actin. And how are we doing that? I told you before that we need the calcium. We need the calcium. Calcium is very important, and now you will know why. Because it binds to troponin complex, troponin, that changes formation, and what it's troponin doing is that it changes also its formation, and it changes for conformation of tropomyosin. Tropomyosin, remember this double helix molecule that is attached to 7 heads of an actin. So we change the conformation of tropomyosin, and this tropomyosin is drawn into the groove between the 2 actin chains. It's drawn into the groove of 2 actin chains, and what it's doing, what is happening is that it's tropomyosin is exposing the binding sites for myosin. So it's like that we have a calcium, so the tropomyosin will move And we'll be like, okay, there you have it. Here's the actin. I was covering it because I'm like shit badass. So now when I have a calcium on my side, you can have it. Take it. Yeah, take it. So it's just like show off the actin. Like here you have it. And that's why myosin can be attached to the actin. Because myosin, when myosin sees actin, is like, whoa, actin, I love you. I need to be attached to you. But it really needs some kind of energy to do so. As you know, you're not probably moving without energy. So myosin is doing the same. It's not moving without energy at all. So it needs ATP. So we need calcium to reveal the binding site and ATP to move the myosin head. And ATP is bound to this myosin head where it's cleaved to ADP and phosphate. So it's cleaved ADP and phosphate because we have ATPase activity on this myosin head. So it's cleaved, yeah. And when the phosphate is released and ADP, when they are released, there is also energy that is released in this chemical reaction right here. And what happens is that the head will move. So now it's attached to actin and it will move. Yeah. So, and it moves in 7 nanometers. Yeah, it will move like to one side. And if there is enough calcium, the myosin head can rebind to the next binding site and the process will repeat. So it will bind, move. Okay? Bind, move. Okay? But it's I'm just like constantly like going to check language sometimes. I'm so sorry. But it will like bind and move and bind and move. But we need enough energy and calcium to do so. So when there is enough calcium, we can move, and we are like a little bit shifting the actin on the other side of myosin. And that's why the sarcomeres, the thin filaments are going between the myosin filaments in the sarcomere. Just try to imagine that as the contraction. When the calcium is depleted, the binding sites are covered with tropomyosin. So when the tropomyosin and troponin complex don't have a calcium, they're like, no calcium, no actin. I'm just covering this shit and you cannot bind. Yeah, so when the calcium is depleted, there is no actin binding site, and the myosin will not bind simply. So that's what's happening here. So remember, we need ATP, we need calcium, we are moving the head, and this is just like the written text that I just described. It's really that we need the calcium. We need to release the energy. Yeah, we need the ATP. And rigor mortis. Rigor mortis is just like the stiffening in the body because of that, death. So it's happening after death. That we don't have ATP because the person is dead. So no ATP here, but still have calcium inside. And what's happening that the tropomyosin is having this calcium, so it's moving and showing the actin side. And you know, when myosin sees actin, it's like, okay, I'm going to attach. Yeah? But really now, what's happening, it's attached, but it cannot move because there is no ATP. It cannot move. And one very important fact that I forgot to say is that when the ATP is cleaved here in the second process, a new ATP molecule will bind, and that will detach the myosin head, and it will be able to connect once again. So binding of new molecule of ATP is very important for the myosin head to detach and be attached once again, and once again release energy, and the process will repeat. So when there is no ATP and is enough calcium, the myosin head is attached, and now it's like, oh, I'm fucked. Yeah, I cannot move, I cannot detach. Yeah. So that's when the contraction occurs and is remaining, because you don't have ATP molecule, new ATP to bind and detach myosin head from there. And it will last a few hours, and after that the calcium levels will drop because the body is dead, so the heads of myosin will detach. And that's what is happening in the rigor mortis, and you will see it during pathology. So once again, yeah, this is just the same ATP, it's like cleaved because of ATPase, energy is released, new ATP will come and it's moving. Just another picture for the same thing. Oh, once again, here's the very really, we have already shown this and how muscle contraction manifests itself on what myofibrils look like. So this is once again the myofibrils, and you can see these bands right here. This is a relaxed state where we have very thick I bands, yeah, and also H band in the middle, because they're not overlapping. But in a state of contraction, the zit lines are moving together because thin filaments are moving to the thick filaments, which are attached in the middle. So this I band is thinner in contraction, also H band is thinner, and everything is very tightly together, connected together. Yeah, because these thin filaments are moving this way to the middle of thick filaments. So this is how it looks like. Muscle contraction is controlled by nerve activity. Really, when we want to contract our muscles, we need nerves to do so. And in the case of skeletal muscle, it's a proper motor neuron which is controlling the muscle contraction. There are very special synapses, they're very special. These synapses are there between the axon and the muscle fiber in skeletal muscle, and these synapses are called motor end plate because they're at the end of a axon of a motor neuron, and they look like a plate, so that's why they're called motor end plate. Nothing special behind the name. And really, you already know from the lecture about nerve tissue that axon doesn't branch. Yeah, dendrites branch, but there is this axon, which is only one axon, and it doesn't branch. But at the end, it branches, and it's creating something called terminal arborization. Terminal arborization, which is just like this, it's branching at the end here, and they are widening on each branch. These right here, you can see these widenings right here, and these are called terminal butons. So we can see one terminal buton or terminal knob here, right here, which contains synaptic vesicles over here. Yeah, synaptic vesicles. We can also see mitochondria here, and these are synaptic vesicles which contain acetylcholine, or like mediators. Yeah, so synaptic vesicles over here. The postsynaptic membrane. I'm going to bring another color over here. So here we have a membrane of a muscle fiber, and this membrane of a muscle fiber is really the postsynaptic membrane. Yeah, you can see it's wavy. That's because we want to increase the area, yeah, where we can bind these ligands in the synaptic vesicles. So they're wavy. There are many receptors here in postsynaptic membranes, many receptors. That's why it's like electron dense structure normally you're going to see. So it's wavy, and we call it a subneural apparatus. because it's under nerve and it's an apparatus, so it's called subneural apparatus. And it's like we can see it here. So what I wanted to say is that the polarization wave, which is transmitting here through the axon, the polarization wave open channels, which then cause the fusion of these synaptic vesicles over here with presynaptic membrane of the neuron. So these synaptic vesicles fuse with presynaptic membrane over here. This membrane belongs to neuron, to axon, is an axolemma actually right here. So the synaptic vesicles fuse with this axolemma, and the mediator is released in a synaptic cleft. Yeah, it's just the space between presynaptic membrane and postsynaptic membrane. This acetylcholine, the mediator, then binds to receptors which are on the postsynaptic membranes, right? Postsynaptic membrane right here. So just acetylcholine binds to receptors and cause opening of the channels, and once again fusion of calcium and sodium, and depolarization wave can propagate or transmit somewhere else. Yeah, so it's causing depolarization wave right here, and the depolarization wave is going somewhere here, and you can see, you already know this structure. This is a T system, or T tubule is this invagination of a sarcolemma down in the center of a muscle fiber, to the center of a muscle fiber. And that's why we have these T tubules, because we want to propagate depolarizing wave, the signal. We want to propagate the signal down to the center of a muscle fiber. Yeah, and that's why we have this motor end play. Yeah, really, this T tubule quickly transmits the depolarization wave deep into the muscle, to the deepest myofibrils. So here we have a nice picture from electron microscope. This is the myoneural connection. This is the motor end plate. I'm going to write it down. It's like a myo, so it's a muscle to neuron, myoneural connection. Yeah, motor end plate. And once again, we can see this is an axon which is widened here, and it's creating this terminal buton right here for this widening. Here we have a nice little mitochondria over here. So this is the really presynaptic membrane. Yeah, presynaptic membrane. I think this is the picture from oral examination, so I'm going to describe it right now. We can see nice mitochondria over here. This is the presynaptic membrane. It's a membrane of axolemma, of an axon. Here we have a postsynaptic membrane, and this postsynaptic membrane is wavy because we want to increase the area where the receptors could be. So for the receptors, everything for the receptors in the cell, like we are going to increase area. So postsynaptic membrane is a membrane of a muscle fiber. There is some kind of artificial dilation, nothing to be worried about. It isn't some kind of special organelle or something. It just happens sometimes during preparation of the slide, so it's nothing to worry about. Yeah, and you can see that. So this is the cytoplasm of a muscle fiber. Actually, this is the cytoplasm of muscle fiber. Really here. This is the cytoplasm of an axon over here, and you can see the receptors over here, these dense, electron dense structures over here that are bound to the postsynaptic membrane. These are the receptors. which are waiting for the acetylcholine. These receptors are waiting because they want to bind acetylcholine here, which is located here in these vesicles, right here. So once again, the depolarization wave is going this way. This depolarization wave causes that the synaptic vesicles fuse with the presynaptic membrane. Acetylcholine is released here, and then it binds to the receptors on the postsynaptic membrane, and the depolarization wave can propagate further to the T tubule. That's an invagination of an sarcolemma. Really, in order for the muscle contraction to repeat in succession, like it needs to be successful. I'm sorry, I'm very excited about this topic, but really, if you want to do it correctly, the acetylcholine must not be bounded in the finally, like it needs to be somehow destroyed, or there needs to be some kind of enzyme which is going to tell like stop, no more acetylcholine here, which is going to cleave the acetylcholine, and that really this enzyme is called acetylcholine esterase, which is here in this cleft right here, the synaptic cleft. Yeah, so there is a space, white space, where normally acetylcholine is transferring, but we have this enzyme acetylcholine esterase, which hydrolyzes acetylcholine, and when it hydrolyzes acetylcholine, it's deactivated in that phase. So, and it's very important, this esterase, that you know about it, that it exists. So, yeah, this is how it works. I struggled with this a lot, as you saw, but I hope you could understand this. So if not, then write down to the comment section that you want to explain it somehow further, or I would try to write down what I meant, but you should know how to describe these basic membranes that are here and what's really going on. Yeah, and these synaptic vesicles over here, and there is also this enzyme acetylcholine esterase, which deactivates acetylcholine, so the like the propagation of the signal isn't like permanent. Yeah, so okay, so let's go next. But we are also able to see motor plates in a light microscope. Like here in these pictures, we can see motor plates also in light microscope in higher magnification, or with like some kind of special staining here. For example, impregnation with the silver. Here we can see a nice neuronized axon which is branching, this terminal arborization, and here we can see these dark spots over here, and these are the plates here, myoneural synapses over here. Yeah, really, one motor neuron innervates more muscle fibers. You can see it right here, one axon, and innervates many muscle fibers. And this is like the it's called motor unit, basic motor unit. So when somebody asks you what's the basic motor unit, you would say, okay, it's like this group of muscle fibers which are innervated with one axon. That's the basic motor unit. Yeah, so one axon innervates many fibers. Here we have a histochemical proof of acetylcholine esterase, which is these enzymes that deactivate acetylcholine, which is a mediator in this synapse. So just a histochemical proof, so we know that we have these motor end plates over here because there is an esterase location. Okay, and this is the picture from, once again, light microscope, and it's very high magnification, so we can see motor plate over here. Nice motor plate. This is the muscle fiber. You already know this. Muscle fiber. And there is the muscle fiber. These are the cross sections of muscle fibers. And we can really see this is labeled with S. These guys over here are Schwann cells. This is the nucleus of a Schwann cell. This is the Schwann cell, like here. And why do we have Schwann cells here? You know, Schwann cells are glial cells of peripheral nervous system. These cells are myelinating axons. They're always around axons somehow. Maybe they are doing these myelin sheaths, but if they are not doing these myelin sheaths, they are just like hugging axons, going with them, and like doing their job, just like supporting them and other stuff. So they're always around axons. So really this is the motor end plate over here and over here, and these Schwann cells are supporting the axons which are here, creating this motor plate. Okay, so that's pretty normal. It's just like part of peripheral nervous system, nothing special going on. Yeah, but we can see it very well. So, mechanism of action. How is this working? I know this looks very scary, but it's very important also in pharmacology. Yeah, so ryanodine receptor and all these receptors are very important. That's why we are discussing them right now. As I said before, here we have this depolarization wave, which was created on a postsynaptic membrane of a muscle fiber. This depolarization wave is going through the T tubule down somewhere where the myofibrils are located. And really this depolarization wave, like, it's further transferring excitation from the T tubule. And here we have the so-called ryanodine receptor located on the membrane of sarcoplasmic reticulum. Yeah, ryanodine receptor located in the membrane of sarcoplasmic reticulum, which is connected with spanning protein, which is here, just like normal protein, which is attached to the ryanodine receptor. It's attached into some subunit here. And it's connecting this ryanodine receptor with a receptor, very special receptor, VDCCL receptor, this one here, which is located on the membrane of a T tubule. And what's happening here, the depolarization wave goes, and then it stops at this VDCCL receptor, and this receptor will open. Because of the depolarization wave, it will open, and there is a slight influx of a calcium ions in the cytoplasm of a muscle fiber. Yeah, so slight influx, and this opening of this channel VDCCL will also open ryanodine receptor. And why? Because we have the spanning protein which moves towards this direction. So when this receptor open, because of the depolarization wave, also the ryanodine receptor will open. And what will happen in that case? When the ryanodine receptor open, the calcium channels that are in the sarcoplasmic reticulum, in the terminal cisterna of sarcoplasmic reticulum here, will go here to the cytoplasm of a muscle fiber. There's very fast influx of calcium ions to the cytoplasm of muscle fiber, because it's this concentration gradient. We have many calcium ions here, very much calcium ions here, so there is very fast influx to the cytoplasm. And now we have calcium in the cytoplasm of a muscle fiber. And why do we need calcium in the cytoplasm of muscle fiber? Well, it needs to bind to the troponin complex. Remember, troponin complex needs calcium in order to change conformation of tropomyosin. So the myosin can bind to actin. Yeah, so that's why we need calcium to activate actin. Yeah, actin is not active. I know it sounds like it's active, but sometimes it's not. Yeah, so we need calcium to activate actin. So, and now you know why do we need so much of a sarcoplasmic reticulum? Why do we need so much smooth endoplasmic reticulum in muscle fiber? Because this smooth endoplasmic reticulum contains so much calcium ions. It's like, yeah, it really contains calcium ions. It's doing another things, like you know the basic function of endoplasmic reticulum, smooth endoplasmic reticulum, but this its function is very important for the contraction because it contains a lot of calcium ions that are very important, yeah, and binding to troponin. So this is really happening right here. You should know the VDCCL receptor. You should know also ryanodine receptor. You will learn about them in the pharmacology, and I hope you can remember them and what are their functions. Really, that's important. And here we have, you can see it right here. Here we have another receptor. It's called the SERCA. Yeah, it's a shortening of an sarcoplasmic and sarcoendoplasmic reticulum calcium yeah, channel. So, and this SERCA is pumping this calcium from a cytoplasm here, is pumping because we need ATP, because it's the opposite way of a concentration gradient. Yeah, so we still have more calcium ions here, so it needs to be a pump here, because you need to somehow control the transportation. This SERCA is always pumping calcium, always. But when there is this opening of ryanodine receptor, yeah, it's not enough for her. Yeah, he cannot, this channel cannot pump all calcium channel outside, so that's why it increases. But when it's closed, when it's this ryanodine receptor closed, in that case the SERCA will pump more and more calcium, so it will lower calcium ions in the cytoplasm. I hope you can understand this. And this mechanism of action, how it's created, is very important. So now you know why do we have these T tubules? Because we want to transmit depolarization wave. We want to open this receptor, so we can open ryanodine receptor on the terminal cisternae of a sarcoplasmic reticulum. So we can have these calcium ions in the cytoplasm. Yeah, but we need to somehow pump them back, because you don't want to contract your muscles like always, all the time. Sometimes it's like you need to end this action. Okay? And you need ATP to pump in a SERCA. Oh, this is a picture from electron microscope. It's very high magnification. It's like, I'm so sorry. Once again, these are authors of this picture say that these are the ryanodine receptors. Like, man, I was surprised also. Doesn't matter, really. Don't have to deal with that. But what about the regeneration of skeletal muscle? Really, regeneration is very not so good. I would say it isn't good. Yeah, when you lose your muscle once because of like you cut it out, it's not you're going to grow your biceps once again. Yeah, cannot do. So it's healing with scar tissue. Really, the skeletal muscle tissue is healing with scar tissue. There is some kind of like very small regenerative ability going on, but it's so small it cannot like build a new muscle. You cannot build new skeletal muscles. We have this dormant satellite myoblast. A satellite is a very strange name because you already know one satellite cells in the nervous tissue, these cells that are supporting neurons, that are around neurons in ganglion, in peripheral nervous system, but also these myoblasts, which are really undifferentiated cells. These are the precursors of muscle fiber, because myoblasts were these cells that fuse together and create muscle fibers. So they are like precursors of muscle fiber, and we have them in very small amount. We normally cannot differentiate them in a light microscope, but we do have them. But it's not like they can form the whole new muscle. It's not happening. Yeah, but we have some kind of regenerative ability, but it's very small. Okay, so that was for the skeletal muscle, and now it's the heart or cardiac striated muscle tissue. So once again, it's striated. It's striated, and we will not take it in such detail. I think that skeletal muscle was enough for you right now. But really, everything we said about the mechanism of contraction and morphology applies also for the cardiac striated muscle tissue, but there are some differences. And there are some differences, like for example, first very big difference, and please remember that, very big difference is that basic unit isn't fiber, basic unit isn't cardiomyocyte. Cardiomyocytes are basic unit, so these are cells. Yeah, nice little cells are creating cardiac muscle tissue. They are mostly cylindrical. They are often branching, so they look like kind of like this. They are branching. Ooh, like this. They have a cigar-shaped nucleus, once again, and the cigar-shaped nucleus is located in the center. That's the difference. It's a cell, it has a nucleus, cigar-shaped, it's in the center, this nucleus. It's cylindrical, it's a branch on the periphery, yeah, and it's a very happy cell, working all day, all night, no rest, really, so that's sad. But you don't need any voluntarily control, so you're not telling your heart to pump, so there are rhythmically contractions. In these cells, they are about 10 micrometers wide, or in length, I should say. 100 micrometers. I don't know if I said 10 or 100, so they're big cells. 100 micrometers. And in diameter, they are like 15 and 20 micrometers. Okay? And I said this already, central position of nucleus. And also they contain myofilaments, which we have described earlier, that are arranged in the myofibrils. So you should know everything that's written here. Right here. They're going to ask you this all the time. And we have here a really nice picture. And these cells, these cardiomyocytes, you can see they're branched, and they are connected together with this intercalated disc. So for example, this is one cell, here's the nucleus of this cell, and this is another intercalated disc here. So we can see it's kind of cylindrical, it's branching somehow. We can see some branching over here. And really, when you look at it in very small magnification, like you can think that, okay, these are the fibers, I can see fibers. Yeah, so they somehow resemble fibers. But really, that's because they are so connected together. They're forming these long fibers, I guess, but these aren't fibers as we saw in the skeletal muscle. This isn't a syncytium. There are normal cells that are connected via these intercalated discs together. And these intercalated discs, these are very special structures that can be found only in cardiac muscle tissue. So you should know their structure and stuff and what's going on really in the muscle fiber. Not muscle fiber, in the cardiomyocyte, which is a cell. I'm sorry. And really, yeah, in your textbooks, I'm really glad it's written here. In your textbooks, like sometimes it's written muscle fiber, yeah, because they're resembling these fibers. But really, just remember these are the separate cells. They are working together, they are connected together, but they're not creating this sensation. Although they're called muscle fibers because they are creating like these connections and long fibers, but really it's not the same as a skeletal muscle. Okay, skeletal muscle is a sensation, cardiac myocyte is a cell, cardiomyocyte is a cell, okay, which is connected together. You should know these differences. So here we have a picture from light microscope. We can see centrally located nuclei right here, centrally located nuclei. Okay, this is a longitudinal section, so it's hard to tell, but here we have a cross section, and here we can see the nucleus in the center of the cell. So it's a different situation. So this is a nucleus of a cell, and this is the cell. Yeah, that's the cell with the nucleus inside, and also this is the cell. So we can see these are very big cells. We can see these intercalated discs. These are these lines. So we can tell this is one intercalated disc over here, and this is the second one over here. So this longitudinal section is a longitudinal section of a cardiomyocyte, and we can see nice cigar-shaped nucleus in the center. We can see some kind of striations sometimes, that's something going on transversely, this striation, but it's not, that's like, you cannot see that well as a skeletal muscle. There is some kind of difference. It's not the same, yeah? So the content, okay, I'm going to tell you right now, why is that so? Is that so because there are also myofibrils and stuff that was also in skeletal muscle, but there are many mitochondria. The content of mitochondria is up to 40% in these cells. Yeah, so there are less myofibrils. They are not connected together that firmly. There is so much more cytoplasm between them with mitochondria, because these cells, yeah, really, these cells need a lot of energy. I can tell you, like 50%, almost 50% of cytoplasm is mitochondria. Yeah, so that's why. And this is the cross section of a cardiac muscle. Cross section. So once again, we can see nice little cells right here. So cells, these are the cells. These are like 15 micrometers thick cells over here, and we can see nice little nuclei in the center, like this. Okay, so this is the typical cross section of cardiac muscle. We can see also lipofuscin, this brown pigment here, lipofuscin brown pigment, because they are living their life all the time. Once the cardiomyocyte will die, it's not able to regenerate, so they're living their whole life with you together. And that's why we can find lipofuscin here. Also, you can find lipofuscin in neurons and that stuff. So remember, myocardial infarction all heals with the scar. Also, it's no regenerative ability at all. Here we have pictures from electron microscope. Once again, here we have the nucleus right here. So we are somewhere in the center. Yeah, so this is the nucleus. It's kind of active. You can see a lot of euchromatin here. It's very active. I mean, like these cells are very active, so nucleus is also active. Here we have the nucleus. here we have mitochondria, because we can see two membranes here, one membrane, second membrane. So I know these are the mitochondria and these are the crysts. So we have mitochondria, and these are the myofibrils. Yeah, the structure of myofibrils is the same as in the skeletal muscle, myofibrils right here. This is a picture somewhere on the periphery of a cardiomyocyte, because we are not able to see a nucleus, but we can see the membrane. Yeah, the membrane, and what we can see here is a cross-section of a myofibril over here. So once again, we can see the thick myofilaments over here, these dense, electron-dense dots that are big, and around them there are thin myofilaments, 6 thin myofilaments around. Oh, there were 7, but doesn't matter. There are 6 around, 1 thick myofilament over here, the thin myofilaments around these thick myofilaments. So this is just a cross section, and you're probably now wondering in which band is it dissect. So here we can see also thick and thin filaments, so it's an A band, yeah. And here you can see it right now very clear. This is very big mitochondria, and they fuse together. So sometimes we can see just very big mitochondria in the cytoplasm, very fused together with many crysts. That means it's very active. When it's big with many crysts, it's very active. So this is the mitochondria, very big. And this one is also here, just to compare these two, they look the same. So this is also the mitochondria over here. They're fusing together. Yeah, so mitochondria, this structure here, cross-section of myofibril. This is a outer membrane. And also, as I said before, there is this superficial lamina also over here. We are probably not able to see it once again here, so we're not able to see superficial lamina here in this picture, but there is a superficial lamina. Yeah, so just remember that. I'm going back to here because I forgot to tell you we were able to see striation, not that good as in skeletal muscle. We can see this intercalated disc, and once again don't forget that muscle tissue is eosinophilic once again, so it's pink, not that pink as a skeletal muscle, it's less pink, yeah, it's a little bit brighter, but it's pink. Yeah, so it's once again eosinophilia here. Here we have another picture, electron microscope. Once again, mitochondria, big mitochondria, almost 40% of the cytoplasm of a cell is about to go to mitochondria. So they're very big, you need a lot of ATP. And once again, there is this myofibril on the one side, on the other side. And you're probably able to tell that this is the A band right here. Yeah, A band, I band. This is the I band over here. Yeah, so it's not very good seen, but okay. So, and what are the differences? Other differences? The sarcoplasmic reticulum is not that big. Yeah, so we can see that we have once again this T-tubule, which is an invagination of sarcolemma, which is once again the membrane of this cell right here. So we have this T-tubule, but this time is on the interface, or it's really located in a Z line here, Z disc or Z line. So it's located here. In skeletal muscle, it was located on the interface of A and I band. Remember, this time we have it on the Z line, Z disc right here, and so here is the T-tubule is wider. You can see it right here, it's wider, and also the sarcoplasmic reticulum here isn't that prominent, so we have a smaller amount of sarcoplasmic reticulum here, and it's not creating this terminal cisternae, only just some kind of widenings of the tubules here, but you can call them terminal cisternae. But what is really happening here, they're not on the both sides really. sometimes when you do a section, when we're going to have a look on a picture from electron microscope and you're doing the section and you want to see, we saw a triad, and if you want to see a dyad at the Z lineage right here, so we're going to dissect it somewhere here, so we'll probably will be able to see this widening of an sarcoplasmic reticulum, this pink thing here, and a T-tubule next. But we're not able to see another terminal cisternae or widening. So that's why it's called a dyad. Not diet, but diet. Like it's like diet, I'm going on a diet, but no, diet, I don't know the correct pronunciation, maybe diet. Like there are two things together. Yeah, that's what I meant. So there's only on one side the terminal cisternae or this widening of a sarcoplasmic reticulum. You can call it also terminal cisternae, it doesn't really matter. So basically there are just fewer of them. There are fewer of them. And there is really one dyad per sarcomere here. So each sarcomere has its own dyad. And the same thing is happening also for the skeletal muscle. What was happening in the skeletal muscle, it's also happening in the cardiac muscle. So the contraction is the same. There is one difference I'm going to tell you, but there is also this binding of an actin, myosin and other stuff, and we need calcium, and that's why we have this dyad right here. So in this picture we can see that this is a picture you can have during oral examination, so now pay attention. What we can see here, and these are of course the ribosomes here that are attached, probably this is the granular endoplasmic reticulum, and here we have a widening over here. This is the T-tubule. It's very wide, it's wider. When you compare two pictures from a skeletal muscle and cardiac muscle, you can see that this T-tubule is wider, and there is an sarcoplasmic reticulum right here on one side, and that's why it's called a dyad. And here it's in the Z line, but we're not able to see myofibrils. We can see, like, probably here we would have a myofibril because I can see these thick filaments over here, so this is just the start of a myofibril, but whatever. This is a very typical picture. When you look at this, you should know it's a dyad in the cardiomyocyte, typical for cardiomyocyte. And what can lead us here is also we have this very big mitochondria, very big, fusing together. So this is the cytoplasm of the cardiomyocyte with the dyad and with mitochondria, and we have probably myofibril over here and somewhere over here. Okay? So once again, the contraction of a cardiac muscle, there are some differences, but once again, we have a depolarization wave that is opening a calcium channel that is going down, down, down here to a T-tubule. And here we have this special connection, once again, this receptor, VDCCL receptor, once again, that is opening channel in a sarcoplasmic reticulum, releasing calcium ions that binds to troponin complex, which change conformation so that the myosin can bind once again and move, so on. And that, after that, calcium needs to be pumped away from the cytoplasm. There is a difference because we're not able to see here this protein which was connecting these two receptors. So we have VDCCL receptor which opens, and there is an influx of calcium channels inside to the cytoplasm, yeah, but we don't have this protein over here. So that means that ryanodine receptor in a cardiac muscle, is really it's opened because of this calcium ion right here. Yes, so we call right now, it's not mechanically controlled receptor, is a calcium controlled or ligand controlled receptor right here in the cardiac muscle. Yeah. So once again, the calcium binds to this receptor, it opens up this receptor, and more calcium will influx here to the cytoplasm, and it's once again pumped back to the sarcoplasmic reticulum by a SERCA. And this type is type 2 because it's somehow different. It's controlled by a protein called phospholamban, which when dephosphorylated, it inhibits the SERCA2. Yeah, so it's just there is some kind of control mechanism which is not very important right now for you, but just know that it's controlled, everything is very controlled, and there is this ryanodine receptor which needs calcium in order to open because it's a calcium controlled receptor right here in the heart. Yeah. So, thus, intercalated discs. We already mentioned them. These intercalated discs are connecting complexes between cardiomyocytes. So here we have one cardiomyocyte and second cardiomyocyte, and these are the intercalated discs. Yes. That's really well described here, like what's happening really in these discs. You can see that we have some kind of cross section of a disc and some longitudinal section. So these are the cross sections of a disc, and this is the C is a longitudinal section of a disc, which is going in parallel with myofibrils. What's happening here is that in the cross sections are made of fascia adherens, which is very similar to a structure you already know from epithelial tissue. That's the zonula adherens. So it's very similar to zonula adherens. Similar proteins are here. You don't need to know differences. You just need to know that fascia adherens is located in intercalated discs, and it's connecting these thin filaments in the myofibrils. So it's really like a Z line. So here we have Z line. It's very similar to Z line. Yeah. So you don't have Z lines between two cardiomyocytes. You have fascia adherens. Yeah. Because you don't want to lose the contraction between the cells. They need to be interconnected very firmly in order to function. So we have this fascia adherens right here, and we also have in the cross section or cross component here we have a macula adherens. This is just normal desmosome, which is here. You already know the structure of desmosome. That's here just to support the connection. Yeah, just to support connection. It's not, there are no thin filaments attached to this desmosome. This is just for the connection. In the longitudinal section we have gap junctions or nexus gap junctions, which are here for the connection and communicating and spreading or transmitting the depolarization wave in the cardiomyocytes. Okay, so and they frequently ask you the structure of intercalated discs, so you should know it. Like what's in the cross section, what's in the longitudinal section here. Here we have a detail. Here we can see cardiomyocytes, and these things over here are intercalated discs. And sometimes you can see it very well, but sometimes it looks like there is only this cross section, but it's really just depends on the section. Here I draw you. Here we have this cross section made of fascia adherens or fascia adherens. Here is this little longitudinal section, and once again we have a cross section. It's very typical for the cardiac muscle tissue. And here we have an electron microscope, and what I want wanted to say, yeah, the intercalated discs. So here are the fibers, myofibrils over here. So these are the myofibrils. We can see once again the mitochondria. They are labeled with C, mitochondria. And these electron-dense structures are the intercalated discs, or this is one intercalated disc right here. So, and it's made of this cross component. Yeah, cross component here is labeled with A, which is a fascia, or how to pronounce it correctly. Don't know, but I'm gonna find it out. And I just labeled it all wrong because it's only this part over here. So we can see small gap here between cells, and it's very similar to zonula adherens. And you can see these fibers, not fibers, I'm sorry, it's too late for me, not fibers. You can see thin filaments going to the fascia. So it's something similar to Z-line, is interconnecting like thin filaments in one cell to thin filaments in other cell right here. So this is the fascia adherens. Then we have a desmosome over here. This is a desmosome, and it's also over here, desmosome. So it's just like dense spots over here. These are the desmosomes, and also I cannot see it properly here, but they're not connecting these thin filaments. These are just the desmosomes, just like macula, dense, electron-dense macula. And the B is the longitudinal section, which contains nexi that are here. So gap junctions over here. We are not able to see them. There are many gap junctions right here together. There's not just one gap junction. There are many of them, many in one place. So this is the structure of an intercalated disc. And here we have a cytoplasm between these myofibrils over here with mitochondria and ribosomes and other stuff I cannot somehow distinguish, but probably some vesicles are there, and I don't know really what else can be seen here. We have also, do you remember costamere? Costamere, this protein complex which was connecting myofibrils together with the outer lamina, superficial lamina, and the membrane of a muscle fiber. Here in cardiomyocytes, it's kind of different. We lack dystrophin. We don't have dystrophin here, which was very important in a Duchenne dystrophy, as I said before. And that's very good because the heart muscle isn't affected in a Duchenne dystrophy because there's no dystrophin here. Yeah? So vinculin in this case is more important, vinculin, yeah, which shares the role with dystrophin in the costamere. You probably cannot see it here because of the camera. I'm going to write it. It's here. Vinculin is very important. Okay, this is the longitudinal section, very high magnification of the ear. And what's it's I don't know, why do we have this picture? Oh, probably because of the capillaries here. We can see very nice capillaries over here. And these are the capillaries. A longitudinal section of capillaries right here with erythrocytes inside. So really what we can see in cardiac muscle tissue, because it needs a lot of oxygen every time, we can see these long capillaries everywhere. It's very typical. You're going to see it during practical trainings. Oh gosh, it's very typical for the cardiac muscle tissue. And once again, we can see the striation just a little bit and the nuclei over here. And there is some extracellular, once again, mass around this cardiomyocyte. And don't forget there is a superficial lamina around each cardiomyocyte. And this is a picture of a cardiomyocyte once again. Here we have nice myofibrils. Yeah, nice myofibrils. There are some vesicles over here, vesicles. And this is the This is the outer membrane of a cardiomyocyte over here, and there should be superficial lamina, but once again we're not able to see it. But what we can see here, there is a little extracellular space over here, and this structure, this is the capillary, and this right here is a wall of the capillary, and this is the lateral process of endothelial cell. Endothelial cells are cells which line capillaries, and this is a process of endothelial cells. I can recognize it because I see many synaptic vesicles over here, because endothelial cells are always transporting something somewhere. But that is very typical for endothelial cell, and you're going to learn about it more right after. But this is a lumen of a capillary. You're not able to see erythrocyte here, but sometimes you can also see erythrocytes, but not in this picture. So, and smooth muscle cells. Yeah, smooth muscle cells. Really, I'm going to find in here because I don't know if I forgot something or no right now, and this is the last thing we are going to discuss right now. Yeah. So oh gosh. So we come to the last, finally the last muscle tissue, and that is a smooth muscle tissue. The smooth muscle cell is a spindle-shaped cell. So once again, we have cell as a basic unit, as in a cardiac muscle. So cells are the basic unit. They are spindle-shaped. They are able to contract very slowly. They usually have dimensions of about 5 to like 500 micrometers, sometimes in a uterus of a pregnant woman. Yeah, because uterus is made of smooth muscle cells, and sometimes really during pregnancy, like always during pregnancy, they're enlarged so much that they are about 500 micrometers big. That's very big. And there is also a central position of a nucleus, and nucleus is once again a cigar shaped, so they look like really like this. This is the cigar shaped nucleus, and with a spindle shaped cell like this. And this is very, very important. The myofilaments do not form myofibrils, so we are not able to see myofibrils in a smooth muscle tissue. Please remember this. This is a very huge difference. And also mechanism of contraction and everything is different than in other. So here we can see that this is the diagram of an arrangement of smooth muscle cells right here. It's like a compact layer. Most of the time it's just a compact layer where it's often difficult to observe intercellular mass or intercellular spaces. We cannot see them here, but there is some kind of intercellular space, and in this space there are many reticular fibers. Probably they're just these things right here, reticular fibers, which play a role in the cohesion of the cells. So we don't have specialized connections, we just have reticular fibers between them which are connecting them. So during contraction, intercellular mass is more involved because there are no special connections or such. There are no intercalated discs, there are no fibers. That's like the whole syncytium. There are just cells which are connected with reticular fibers, creating a compact layer like this. Yeah, with cigar-shaped nucleus in the center and like spindle-shaped. And this is a picture at a small magnification we can see it. Sometimes somehow it looks like this, complete mess. But this is the normal, this is how it looks like, the smooth muscle. They are forming these bundles. These are many smooth muscle cells forming the bundles, really. So, and we can see these are the vessels over here which are penetrating to the muscle tissue. So everything here is the muscle tissue right here. Maybe there are some kind of septae. These are some septae which are made of connective tissue that are between muscle tissue, like here. We can see some septae, but these are the bundles of smooth muscle cells. So, and this is very high magnification, a longitudinal section of smooth muscle cells, and we can see a nice cigar-shaped nucleus over here, but you can see they look kind of strange. No, you can see it. They are not just like normal nuclei. They're during contraction, they are somehow rolled, and we can see them as, we can see these contractile nodules. So during contraction, cells just like, it's a little bit twisted. It's like crunched everything together. We can see these nodules, and it's this is a cell in a contraction, really smooth muscle cell. So we can see the cytoplasm around, which is once again eosinophilic. Cytoplasm in all muscle elements is eosinophilic, and they are spindle shaped, and they have this strange nuclei. When they contract, they are like twisted or somehow like crashed together. You will see in another picture. This is a picture from light microscope. We can see longitudinal sections of smooth muscle cells, and once again they look like really fibers right here. But remember, these are the cells, many cells together. You're not able to tell really the borders between these cells in the longitudinal section, but here is the cross section, and you can tell these are the separate cells with nuclei in the center. These are the cells. And sometimes, more of the time, we're not able to see the nucleus, because I'm going to write it down. This is a nice cell which is spindle shaped, and you have many sections. You can dissect it here, here, so you're not able to see a nucleus, yeah, in the cross section. Like here, there is just a part of a smooth muscle cell, but when you dissect it right here across the nucleus, you're able to see the nucleus right here, which is round in a cross section, obviously. So these are the separate smooth muscle cells, and they are creating bundles. They are together, working together, yeah, like this. So these are the big bundles of many muscle cells, smooth muscle cells. And this is how it looks like in an electron microscope. So we have light active nucleus here in the center. We have a membrane over here, which is once again surrounded by a superficial lamina. Like always, I'm repeating myself, I'm so sorry, but you need to learn this stuff. And this, we can see what we can see inside. That we are not able to see myofibrils. There are no myofibrils here. What we can see here is these like lines or something is going on in the cytoplasm, and these are thin filament, many thin filaments. So actin is here forming filaments. Yeah, so there are no myofibrils, but there are filaments here, and these are thin filaments. Probably now you're asking, and thick filaments? Are there any thick filaments? So just wait. This membrane over here is creating these invaginations. These are called caveoli. They are just enlarging the surface. They are somehow like T-tubules. They are not so deep. There are like many T-tubules close together. Caveoli. And they're for the same thing. They are transmitting depolarization. Yeah. We can see ribosomes over here, this dot. So what we can see here, the nucleus, the cytoplasm with thin filaments of A-actin. We can see caveoli, these small invaginations. And what else can we see are these dense spots over here. Dense spots. And these dense spots are resembling probably Z-line or something like that. They are not Z-line because we don't have myofibrils, but they have the same function. They are supporting, they are connecting thin filaments in a muscle, smooth muscle cell. So also they contain an alpha-actinin, which also can be found in a Z line in a cardiac muscle or in a skeletal muscle. So I'm going to write it down like alpha-actinin once again, which is connecting thin filaments. So here we can see how does it look like. This is the relaxed smooth muscle cell, and this is contracted, so it's like twisted. And here we have these caveoli, which are invaginations of a membrane right here. Yeah, so these were the dense bodies, which were connecting thin filaments together. So dense bodies over here, these guys over here, thin, connecting actin filaments. Everything is curved somehow. Here we can see it once again. Nice membrane of a smooth muscle cell with caveoli right here, invaginations. And under membrane here we have these dense bodies which are connecting filaments. It's like this. Dense bodies over here. And many lines. These are the thin filaments. But remember, no myofibrils at all. This is the mitochondria over here. Once again, you can see the whole network of these dense bodies. Yeah, so this is a very nice picture. Many muscle cells over here and many dense bodies. So you're probably still wondering where's the thick filament. Yeah, here once again we have the invaginations, the calveoli. Okay, that's very cool. Very cool. Here we have another membrane of another smooth muscle cell. So here we have also caveoli over here. Yeah, some kind of elastin over here going on. The extracellular matrix is here, extracellular mass. And this is a special staining or impregnation that visualize the reticulin fibers that are between the muscle cells over here. Yeah, so that you know that they are important for the contraction of a whole bundle of smooth muscle cells. And we have also some kind of contractile non-muscle elements, and these are very similar to smooth muscle cells. So we have these myoepithelial cells that are in glands which are contracting, so you can release the secretion of the gland. Yeah, we're going to describe these cells further in our practical trainings or lectures. So just now, it's not very important to talk about them, but write them down, and right after you can repeat them. Okay, so they are these non-muscle elements that are very similar to smooth muscle cells, so they are able to contract, and they probably look somehow similar to them. Okay, so these cells. Yeah, these are the pictures, but right now I can tell you it's not that important to recognize them, so we're not going to stay here. It's really not so important. But what is important is the contraction of smooth muscle, because it's the same in smooth muscle and non-muscle elements or non-muscle cells which are contractile, that can contract. Really, here is the myosin thing. Yeah, you saw actin, the thin filaments, but myosin is not arranged in myofilaments in a smooth muscle cell. The myosin is inactivated in the smooth muscle cell, which is not contracting, is inactivated. So these heavy chains of the myosin molecule are somehow like twisted. You can see it right now. They are twisted. And really, in order for a thick filament to myofilament to form, once again, in order for the myosin molecule to form a thick myofilament, it needs to be stretched, and it's stretched because of the phosphorylation right here, which is provided by myosin light chain kinase. And what do we need for the phosphorylation is an ATP, so we need energy once again, and we need calcium calmodulin complex. So first we have this calmodulin, which binds to calcium, and then this whole complex binds to, like, will activate the phosphorylation of myosin molecule, because we are going to activate the myosin light chain kinase. Kinase stands for phosphorylation. When it's somewhere is written kinase, it's going to phosphorylate some structure. It's stretched, and when it's stretched, the myosin molecule, it can form thick myofilaments like here, these. And now when we have thick myofilament, we can somehow bind to an actin in the cell and somehow stretch the smooth muscle cell. Now you think, like, why is that important? Also, once again, it will be important in pharmacology. Yeah, in pharmacology, very important. Yeah, so we have some kind of inactive state, and when it's phosphorylated, it's an active state. So applying that to muscle tissues that we were talking before, in cardiac muscle and in skeletal muscle tissue, there was actin that needed activation. Remember troponin complex? You cannot find troponin complex here, so you had to activate an actin. Here in a smooth muscle, you need to activate myosin. Myosin is the structure that needs calcium, yeah, in smooth muscle cells. That's the difference. And really, this interaction, once again, is perceived just as in a cross-striated muscle, or the skeletal muscle and cardiac muscle right here is the same. Yeah, so it's just a picture from here. It's the polarization wave that open calcium channels. Yeah, here we have a calcium gated channel in sarcoplasmic reticulum. So once again, we have this ryanodine receptor here. Once again, but this difference is that the calcium isn't binding to troponin. The calcium is binding to calmodulin, forming complex, and then activating the myosin light chain kinase, activating myosin to form thick bundles or thick myofilaments. Okay, so that's the end of my presentation about muscle tissue. I think I said everything, and you need to know, really, like, if you want to know the basics of the muscle tissue. I know it sounds scary, but you should know these things, and some of them are more important, some of them are less important for you to pass exam, but you should know the mechanism of contraction. That's very important in your further studies. You know, as I said, in pharmacology, you should be able to tell where is the mechanism of action, or why do we need these drugs for that and that, and you should know the basic differences between elements. Yeah, what's the basics of skeletal muscle tissue, cardiac muscle tissue, the smooth muscle tissue? How can we recognize these in light microscope? Yeah, and how do we describe them in electron microscopy? I hope this presentation was clear for you. If not, please write in the comment section questions or in the email, and I'm done. Okay, so bye bye, see you next time.